Okay, so here we are throughout the program. We're now going to get into the microbiome and specific applications. And the goal of this is to kind of show you some of the research with the microbiome, how it impacts various health issues, but more importantly, to talk about how the microbiome impacts health and all the key mechanisms that are involved with improving the richness and diversity of the gut microbiome. So remember, ultimately, when you're trying to treat the gut, to improve health, you're trying to impact these different gut axes. The gut-brain axis, the gut immune axis, the gut endocrine axis, the gut skin axis, the gut lung axis, all those really depend upon a diverse, rich microbiome. So at the end of the day, when you really look at how digestive disorders cause health problems, we know that they cause health problems when you can't digest, absorb the foods, but ultimately, you have to have a very healthy, diverse microbiome with lots of richness and resilient to stressors. That's the key thing. So it's pretty clear in the research the past 10 years, if you have things like endotoxemia, you're in trouble. If you have things like leaky gut, you're in trouble. The other key thing is microbiome diversity and how rich and diverse your microbiome is. So the key thing, though, with the microbiome diversity is if you really look at different profiles and panels with individuals, it's not easy to necessarily change, even though studies show they change within 2 to 3 days, especially if there's underlying issues. So what I'm trying to emphasize throughout this whole program, because here's you guys, I know everyone knows here how to use enzymes, and everyone knows here how to what leaky gut is. I know all that. The problem that we see is the priority of hierarchy, not understanding how important endotoxemia is, and then trying to jump into treatments where it's not going to work. Okay, so the thing that comes with the gut, it's really a hard topic to teach because everyone already does it and everyone is involved in doing lots of stuff. So, you know, the problem is when you really take a look at why gut issues don't heal, like meaning, for example, for myself, my biggest growth comes from my patients, like most of you, and what I do call case reviews. So for me to take on and work with a new patient, yes, I still practice, by the way. I keep getting asked, yes, I practice. I'm not all theoretical, I actually practice. Okay. So in real practice, I get case reviews. I get to see exactly what happened. I get to see what was done with another practitioner. Most of the time, I'll see the people that they're working with and go, they're really talented. Like they had a good workup. And it's the slip of not understanding the hierarchy that makes all the difference. It's not really figuring out what type of leaky gut. It's the jump into protocols where things go wrong. It's the over-supplementation without understanding the mechanism. Those are the key things. And because a lot of times what successful clinical management is you understand the pathophysiology and you teach it and explain it to the patient. Like you don't just have leaky gut, you have leaky gut because you have autoimmunity, and this is going to be an ongoing issue for you, and you have to constantly work with this. Just the way it is. It's part of the disease. And if you can understand that, then it's different. Then you have a patient that really can deal with what they have instead of feeling like they have some rare disease, no one can figure out what to do, and they don't understand how things work. Okay? So we talked about digestion. We started to get into the immune function. We talked about intestinal permeability and tight junctions. We need to talk about immunology a little bit more as we jump into the microbiome. So before I jump into the whole microbiome part of it and all the things that impact diversity of the gut, which is important for all of us to do the best we can, is to really get into this concept of immune reactivity in the gut. Now, if we look at one thing, which is adverse reactions to food, we can have adverse reactions to digestive enzymes, right? We talked about that. So people that can't tolerate protein, think of hydrochloric acid. People that can't handle starches or carbohydrates, think about pancreatic enzymes. People that can't tolerate fried or fatty foods, think of gallbladder, bile release, right? People who can't handle portion size, think of dysautonomias. People who can't handle any foods, think of small intestinal bacterial overgrowth. So those are mechanisms. Another mechanism where people have intolerance to food is they get intestinal permeability, and food particles cross. And when food particles cross, they develop food sensitivity. And then they develop food sensitivity, then you have to deal with the permeability issue. But another deeper mechanism is loss of oral tolerance. And oral tolerance research has been around for a very, very long time. It is just not translated through the world of functional medicine and alternative medicine. Like oral tolerance research goes back 30, 40 years. Just some disconnect happened. Like everyone fell in love with leaky gut and forgot about oral tolerance. Okay. Now, tolerance is just your immune's reactivity to the environment and to yourself. Oral tolerance is specific to food proteins, so they also call it dietary protein tolerance. Okay? And one of the things that we need to get into is how oral tolerance works and how immune activity functions. If you want to evaluate your own health, here's what you need to have. You need to have no digestive issues. You need to have no symptoms of hypochlorhydria, pancreatic enzyme deficiency, gallbladder problems. Everything has to be fine. You should have a intestinal permeability test, have no permeability, and especially no endotoxemia. And if that's all under control, you got to make sure you have tolerance so you're not reacting to every food protein, and then your microbiome is as diverse as possible. If you can do that, you're going to be in good health. Okay? If any of those things are defective, then you got a problem. Now, if you're dealing with people that have chronic inflammatory, immune, or chronic diseases, somewhere along that chain, you might need to figure out what to do, right? And figure out what mechanism you need to support. So let's focus really on this concept called oral tolerance. And this is, let me tell you when you know oral tolerance is an issue. You do a food sensitivity panel, and almost every food is positive. Okay, that's a loss of tolerance. When everything they eat causes a reaction, that's a loss of tolerance. And sometimes it's very obvious because they tell you, and sometimes it's not so obvious until you do labs and you see everything show up. Okay? Now, intestinal permeability is part of losing your oral tolerance, but it's not all of it. There's other things that are involved. That's what we want to talk about. In the world of immunology, there's a concept called anergy, which is a lack of response to an antigen. This is what you want to achieve with dietary proteins. You don't want the immune system to respond to food proteins adversely, have an overzealous response. So in order for this to happen, protein antigens have to be broken down by digestive enzymes. Again, north to south, same thing with immune tolerance. The protein antigen has to be taken up by Peyer's patches, and the immune response, Treg cells, have to not react against them. Okay, and that's like the basic concept. Now, if you look at this diagram, this is showing you all the different degrees of permeability. It's first of all showing you a normal barrier response. You have healthy commensal bacteria, you have mucins, you have SIgA, and then you have some permeability, which we talked about, some minor defects, into significance. This is transcellular, here number 4, transcellular destruction. You can see the gut lining destroyed. And then you see minor defects, that's pericellular, number 3 is in between them. This is a summary of all these things that are there. But you see the stuff below, the mucosal response, the Treg response. I can move it. Okay. When you see those responses, then this is the area I want to focus in right here. So immune tolerance. So if you look at this diagram on the left-hand side, do you guys see Ag? That's an antigen. Just say that's a food antigen. Okay? You have cells called M cells in the gut. What M cells do is they take up these antigens and expose them to that are called dendritic cells. Dendritic cells are Pac-Man cells. They sample everything. These dendritic cells can be overactive. And if they are overactive, you get some serious issues with food reactivity, immune reactivity to everything you eat. And these dendritic cells also turn on brain inflammation, and you get this cascading event. Now, if you take a look up here, you can see SIgA. Do you guys see that? Secretory IgA. So SIgA levels are there, and they're just immunoglobulins. But one of the things that SIgA levels do is they surround food, and when food particles attach to SIgA, like you can see here, SIgA attached to a food particle here, then the dendritic cells no longer sample them. They kind of protect the overexposure. So one of the things that happens when people lose tolerance is they lose their SIgA count. When their secretory IgA count drops, they start to have overactivity of dendritic cells, and they get an overzealous dendritic response. Now, secretory IgA levels can be measured in saliva and in stool. I will tell you what I've learned recently, this past few years. I think it was 3 years ago. The labs that measure hormones that all measure SIgA all do it wrong. Because they're always what? Negative. If you're looking at SIgA through the same lab you're getting your hormones measured through, guaranteed it's going to be low. That's not reality of patient population. That's inappropriate laboratory testing. Because you have to have your reagents to stabilize SIgA count, which you don't do with hormones. So For example, if you're getting the general salivary profile and they measure SIGA and it's always low, that's not real. That's bad, bad technology. The labs should be ashamed. They should stop doing that. It's giving people false information because they're not doing the test appropriately. SIGA needs specific reagents to stabilize them. So if you think SIGA is low on everyone, it's because you're getting the wrong analysis. It's not low on everyone. Okay? Now, SIGA has to be done as its own test with own reagents, not to be thrown in with a hormone collection. Okay? Now, not everyone has low SIGA. I know, like I said, I work with Cyrix. I go, what percentage of your people that you get tested with SIGA actually are clinically low with proper reagent stability? And it's like 20%. So if you're getting 100%, that's part of the testing. Okay, now when people have low SIGA, they can lose their tolerance. Okay? So we know that SIGA is a key factor. And then we know these dendritic cells. These dendritic cells can totally dysregulate if they don't have enough signaling agents, short-chain fatty acids, butyrate, microbiome, polysaccharides. They all metabolize and activate dendritic cells. Those are critical. And then once these immune cells get through the gut, you have cells called, instead of just DC dendritic cells, you have T cells and Treg cells, and they also modulate the immune response. And if these cells are dysfunctioning, you get inflammatory immune responses in the gut. Again, unrelated to leaky gut. Their own distinctive special mechanism. So these things, these secretory IgA cells, these dendritic cells, are all part of normal physiology that get overlooked, that overshadowed by leaky gut. But they're all clinically relevant and important. Now, when you look at protein particles, when antigens come in and they have the immune system react against them, they get taken up in Peyer's patches. You can see those in histology slides of the gut. And the mesenteric lymph nodes take them up. And here's the image here, MLN, mesenteric lymph nodes. Here's the Peyer's patches, PP. They get into peripheral lymphatic nodes throughout your thoracic, throughout the rest of your body. And they circulate throughout your thoracic duct, and they all become systemic very quickly. So undigested food particles, LPS, all these compounds immediately get into the systemic circulation through the mesenteric lymph nodes. Okay? And this is really an important pathway. And by the way, you see these arteries here, there's arteries within the gut called the mesenteric vascular system. If there's hypoperfusion, like someone has congestive heart failure, or someone has significantly low blood pressure, none of this works. And I'll show you some studies about that later. But these are critical. I'm sure about some of those studies tomorrow. So when you're looking at the immune function of the gut, first thing you have to really remember is it's more than leaky gut. Dendritic cells, SIGA cells, Tregs are key players as well when you look at the gut. So this is a diagram, and we show this diagram and talk about how the gut immune tolerance system works. And actually it starts with saliva. So in your mouth, everyone has dendritic cells also. So there's SIGA and dendritic cells in the mouth, and they immediately communicate with the gut microbiome. So there are messenger systems in the mouth that directly communicate to the gut microbiome. So you're already having an immune response to foods as soon as you taste them. You don't even have to swallow them. The immune response is happening, okay? And then your food particles are immediately being surrounded as you chew them by some SIGA, and there's some digestion taking place, and this whole system is starting, okay? And then as you chew and you swallow, you're actually activating the vagus. And as you activate the vagus, you're causing autonomic change. You're getting blood flow to the gut. You're getting sympathetic and parasympathetics under control. You're getting an autonomic function of the gut that controls the immune system. There's a concept called neurogenic inflammation. When you have dysfunction in the autonomics, you can get inflammation in peripheral tissues, and you can release things like substance P. So you have to have proper autonomic control to really improve tolerance. And then food particles have to be broken down. And this is back to north versus south. And when you look at it from an immune perspective, remember that you can't have an antibody response to an individual amino acid. So as long as the food protein is still a peptide, you can have a reaction. So let me show you this diagram here. This is, let's say, an undigested food protein. Okay? I don't know, let's say it's carrot. something. On the left-hand side is a normal response. Digestive enzymes come in, they take this amino acid, this protein, they break it to a polypeptide, smaller version of proteins, right? Smaller chain. This polypeptide gets broken down by things like hydrochloric acid to individual amino acids. You see that? Each letter is an individual amino acid. So think of each letter as an individual amino acid. Peptide is a cluster of them, and a protein is a whole bunch of different clusters of letters. Once an amino acid is developed from the breakdown, there's no immune reaction against it. Everything's good, even if they had leaky gut. If you have an undigested food particle and you have lack of enzymes, these polypeptides are there. Antibodies can attach to peptides. They can attach to an amino acid. So even more so, if you have intestinal permeability, you have to make sure you break down proteins. That's the key thing. Like, it has to be broken down. So this is essential for immune tolerance. Like, you will have people, if you know your patient has uncontrollable leaky gut that's going to take a long time to heal, you better make sure they're digesting and breaking down their proteins. Because if they don't, those polypeptides are going to cause antigenic change. Do you understand? So, like, hydrochloric acid, digestive enzymes are really, really critical for ongoing leaky gut. Because they cannot afford to have peptides get exposed to the gut mucosa and get absorbed. They can afford to have amino acids. That's not going to cause an immune response. But peptides is a whole different story. So digestion and breaking down of things is a critical part of immune tolerance. And I'll be honest with you, if you're not sure if they need them, just give it to them. Because if you have someone who's got autoimmunity or immune dysregulation, just make sure you at least take apple cider vinegar, take some hydrochloric acid, take something to ensure protein breakdown. You can use different enzymes. Now, then you have the dendritic cells. Once the food proteins get broken down to amino acids or some peptides are left, dendritic cells and SIGA are involved, like we talked about. SIGA wraps around them, so all these dendritic cells don't get overactivated all the time. Dendritic cells sample them to decide whether to create an immune response against them or not. Here's the dendritic cells. Again, food samples come in, they get to the gut, they either respond or don't respond. They eventually get into circulation, and you get antibodies produced. And then once these food particles go through the intestinal, the dendritic cells, then that's where you have intestinal permeability, which we talked about. And once food particles get through, you have T cells in the gut. And the most important T cell is a regulatory T cell. And dysfunction in regulatory T cell will cause chronic intestinal inflammation, unrelated to intestinal permeability. Now, if you have intestinal permeability plus dysregulated T cells, you're in trouble. Okay? And regulatory T cells balance out whether to have an exaggerated immune response against food proteins or to not have an exaggerated immune response. And these are the things we know that impact Tregs. The diversity of the microbiome has an impact on Tregs. Because post-bacteria signaling agents impact Treg function. The less diverse the gut is, the more Treg activity you get. Less Treg activity you get. Exercise, healthy relationships, opioids have a tremendous impact on the gut microbiome. So people that are happy, people that exercise, they get opioid releases, they change Treg function. Vitamin D is important. Short-chain fatty acids, butyrate, acetate propionate are important, and glutathione. These things are really critical for immune tolerance issues. So if you guys see a patient that comes back that has like all these food reactions and reactions to food proteins and all these antibodies to foods, but they don't have leaky gut, you may want to focus on Treg cells. Okay? And then food particles that get ingested, they end up getting into the hepatic circulation as well. And here you can see, for example, the portal vein goes right from the gut right into the liver. And within the liver, you have cells called antigen-presenting cells. And antigen-presenting cells will then have a response and create an inflammatory reaction or not. Now, the pre-existing state of this liver, how inflamed it is, how activated these Cooper cells are, is going to determine tolerance too. Because once these cells get activated with an inflammatory response, they can impact the gut microbiome in a bidirectional way, cause inflammation. So chronic chemical exposures, chronic activation of the hepatocyte pathways can also lead to microbiome inflammation. It's a vicious cycle. Okay. Now, this is all called immune tolerance. Okay, this is all part of the immune tolerance pathway. We're going to talk about this a little bit, and I'm going to give you some protocols with it. The key things for each of these pathways, like for dendritic cells, SIGA cells, short-chain fatty acids, A and D, you got to take digestive enzymes to break things down. For the regulatory T cells, glutathione, short-chain fatty acids, A, D are critical nutrients, and then just detox pathways being effective or important there. And when you look at that, those are the main supplements, but that's not enough. Okay, those are just the things you can use to support them from a nutraceutical point of view. The most important thing about tolerance will be diversity of the microbiome. Okay. Now, I have a list of all those supplements and protocols for you. I'll give you in a second. I do want to share something that I just finished with Elaine. We created a 3D immune tolerance program, and the 3 Ds of tolerance are diversify, distinguish, and downregulate. Those are the main concepts. So when you're looking at immune tolerance, you want to diversify the microbiome, provide fuel for the microbiome, improve intestinal antioxidants. Those are key things for diversity. Remember that study where they showed running destroys the tight junctions? So when you do high amounts of oxidative activity, whether it's working out or exercising, being exposed to chemicals, eating foods that are very oxidative, like fried foods and stuff, you destroy the microbiome. So one of the key things that helps the diversity of the gut is the gut having plenty of antioxidants. Okay? The gut has to have fuel, fiber, short-chain fatty acids are critical, and has to have lots of different plant fibers. Those are all critical for the microbiome. Okay? Distinguish, this is where Treg cells come in. We talked about those a little bit. And then downregulate is making sure the liver doesn't have inflammatory responses, making sure you have proper biotransformation pathways. And we created this program that should be out soon. But we worked with a chef. We created all these microbiome diverse diets and all these things just so I can teach the public how to immediately jump into supporting their protocol. But the concepts are just what we're talking about. 3, diversify, get the Tregs to work, make sure they don't have intestinal permeability. Those are the basic concepts, okay? When you're looking at these supplements, if you want to support them, this is the key thing right here. Number 1 is short-chain fatty acids. Glutathione, intestinal antioxidants, DA, digestive enzymes, because they got to break down those proteins to polypeptides. And probiotics are the least effective. I mean, they're effective, but they're the least effective. Okay. Now, the big thing to remember, though, is that microbiome diversity is essential for restoring oral tolerance. The biggest mistake we see clinicians do is they put a patient on a very restrictive diet, like the intestinal permeability diet, the comprehensive one, autoimmune paleo. And when the patient gets on the autoimmune paleo, because of frustration, because of lack of creativity, they make the same meal every day. And that same meal every day is going to limit the diversity of their microbiome. So we tell patients that are dealing with microbiome diversity, you should be eating things every day you don't even like. Do you like Brussels sprouts? No, I hate them. Perfect, eat them. Because the things you don't like are the things your microbiome's not getting. Does that make sense? You could even say, tell me the top 10 vegetables you don't like. Great, make sure you get those in. Tell me what you love. Limit that. And that's how you start to diversify the gut microbiome. Now, some of you guys have heard me say this, but one of the things we like to do is make a mash. This is one of the things in our 3D immune tolerance program will be online. If you guys are interested in it, it'll be available online if you go to drknews.com, maybe in the next month or so. But it's the goal with teaching recipes and a step-by-step approach to help improve it. It's very basic. It's not as advanced as this. And in the autoimmune module next course, we're going to take such a deep dive into this. It's going to be a whole different world, okay? But right now we're just talking about digestion. But when you're talking about one of the things you can do is you can take vegetables and put them in a Vitamix, like I have my patients do. We call it the mash now, vegetable mash. We'll have them go to the health food store, get as many different vegetables as possible, break them all up, cut them all up, because if they get like 30 vegetables, and they try to make like a vegetable drink, it's going to be this big. They're going to have like a gallon and a straw, and they're not going to finish. So you cut it, put it all in Tupperware, have them grab a handful, put it in like a Vitamix with some water or maybe juice or whatever, and then they drink it. And that's just the best protocol supplement you can give them. Lots of different diversity, lots of different plant fibers, okay? And have your patients take a picture of it and send it to you. That changes everything. Hey, when you figure out the mash, send me a picture, email it to me. I want to see what it is. See what happens. And then you'll see the ones that actually do it. And then those are the ones that you can really make sure you're getting something to really diversify their gut. Okay, now, remember, it's very hard to diversify the gut, even with plant fibers, if there's hypochlorhydria. It's very hard to promote diversity of the gut, even with the vegetable MASH diet, if there are lack of bile salts activating these gut pathways, right? So remember those things. If you have chronic inflammation in the gut, it's going to have limitations how much diversity you get from your system. If you're constantly getting exposed to smoke and BPA, microbiome diversity also is impacted. So you still have to look at all these factors. So it's not like you just have this drink and everything's magical. It's just one way to assure you have precursor sources to help the bacteria. But if the pH is off because they have hypochlorhydria, or if they don't have if they have too much inflammation in their gut, you're going to have limited degrees of diversity. Okay? And remember, we have less diverse microbiomes. You know? So, what impacts microbiome diversity? What have researchers found? And what does the microbiome really do? That's what I want to go over with you. Okay? So first of all, the microbiome does many things. It actually contains phase 1 and phase 2 biotransformation enzymes, just like the liver, to detoxify chemicals. The more diverse bacterial species you have, the more potential you have to metabolize a list of different chemicals. Some bacteria will clear out BPA, some bacteria will clear out formaldehyde, some bacteria will clear out different toxins. So the key concept of diversity is to have all these bacteria that all have different enzymes and different potential. Some will do sulfation, some will do acetylation, some will do glucuronidation because of the enzymes they can take, just like phase 2 detox pathways. So different bacterial species are involved with different types of metabolism of end products, toxins, and so forth. So that's one of the key points of richness, is that you want to have all these different bacteria to metabolize as many different chemicals as possible. It's not just about the liver. Like your liver has phase 1, phase 2 pathways, and you can give them precursors like sulfur and things to help them and B vitamins, but your actual microbiome, the diversity you have there, is totally dependent upon your diet and environment. So you can actually change it, and you can lose it. Okay? Now, important point. Take a look at your own diet and see if it's the same every day. If it's the same every day, you're in trouble because your diversity is going down. Okay, you want to expand it. Now, bacteria have the ability to metabolize botanicals and flavonoids to active compounds. We talked about this in last course with different flavonoids for the brain, inflammation. We went through those, but different bacteria metabolize most botanicals are not in their active form. They get activated by bacteria. Right, we talked about curcumin and resveratrol and different types of flavonoids, pomegranate. They become active only when gut bacteria metabolism to anti-inflammatory end products. Then they have an effect on tissue. So the more diverse the gut, the more ability they have to respond to anti-inflammatories you give them. And this is one of the things we talked about. If you give someone flavonoids like, turmeric or, not turmeric, like akashi or blueberry extract or any of those things, pomegranate extract, your effect is limited based on the gut microbiome diversity. But then also studies show, if you give them those things, you start to help diversity too. So it may take a time for them to have an anti-inflammatory effect. Okay? Some patients you give pomegranate extract to, significant anti-inflammatory effect right away. Others, no effect. Part of the reason could be their microbiome. And they can actually make those end products. Bacteria have the ability to manage metabolic modulation energy. This is a major area of diabetes research right now. Most of the papers you guys are, if you look in the diabetes journals, are about the microbiome now. Because it's such a shock to them that this is where they're focusing all their energy efforts into. Because they're looking at the microbiome to fix diabetes and treat obesity. This is a field called immunometabolism, microbiota metabolism. And it's fascinating stuff. And you have to have diversity of the gut bacteria to make these G-coupled proteins and energy signaling pathways work. Gut bacteria also have post-translational modifications. So you have proteins, they bind to a receptor and they have a proteomic response. So these proteins have what's called a post-translational response based on gut microbiome gut bacteria, changing the protein so they can actually bind to a receptor with their own protein system signaling pathways. So they're absolutely critical. Gut bacteria metabolize hormones. If you ever look at a urinalysis profile, you ever see a urine hormone profile, like a Dutch test or something like that? Those hormone levels that say 2-hydroxy and 2-ethyl and all that stuff, that's all metabolized by gut microbiome. Those aren't actually even produced by the endocrine gland. Endocrine gland makes hormones like estrogen. Everything you're seeing on those urinary profiles are made by the gut microbiome. Okay? And then gut bacteria have the ability to develop cellular messengers. And that's important. So it's critical. Like, again, this goes back to, like, what do you do? But remember, you got to start north and go south. You got to make sure those other things are working to really improve the richness. Smoking, we know, causes intestinal permeability, but smoking also causes decreases diversity of the intestinal microbiome. And this includes secondhand smoke. And it's going to be the same with other environmental pollutants. Not that cigarette smoke is special. It's just environmental pollutants will have a change in the microbiome. And people who live in very polluted areas like Mexico City, which is highly polluted, they have very restricted microbiomes. So despite what you eat, the environment you're in can have an impact on your microbiome. This is the effects on antibiotics. So antibiotics have, short-term antibiotic treatment is able to shift the gut microbiota to long-term alternative dysbiotic states, which may promote the development of aggregation of disease. Common features of post-antibiotic dysbiosis include loss of taxonomic and functional diversity, combined with reduced colonization resistance against invading pathogens, which harbor danger to microbial resistance. They really cause diversity issues. Everyone is always open to, you go on antibiotics, take a probiotic. But actually, when you go on an antibiotic, you should diversify your gut. You should really be taking short-chain fatty acids and a vegetable mash with lots of different vegetable fibers to get the GI tract back. A probiotic is okay, but you want to do more. Okay? Here's a study where they show healthy microbiome resilience. Antibiotics come in, they destroy the diversity. You see how diverse the bacteria is here on this left-hand side? In the middle, what you're seeing is less diversity. Once there's less diversity in the gut from this antibiotic approach, it may stay this way. So you want to optimize that change, and we know that studies link dysbiosis to everything from obesity to cancer to autoimmunity to susceptibility to infections. So again, probiotic is probably not enough. You can do more. So promote vegetables, like a vegetable mash drink, lots of short-chain fatty acids. Those are the most important ones for diversity. And the reason I say short-chain fatty acids is why, because short-chain fatty acids like butyrate, they become the fuel to allow bacteria to populate. So when you eat fiber, bacteria make short-chain fatty acids. Those short-chain fatty acids are then used by the surrounding bacteria to fuel their production and their diversity. So when you actually take oral short-chain fatty acids, you're actually immediately giving them fuel. So when you eat a vegetable drink with different fibers, you're not going to give them the fuel and the fiber source to create new bacteria. Okay? This is a study on glucometabolic alterations. Our study is the first to provide evidence for sleep deprivation induced change in microbial families of bacterial gut species, which have previously been linked to metabolic pathologies. So lack of sleep impacts microbiome diversity. You should go to bed by 10. I should go to bed by 10 too. I'm nervous. I had to do a presentation. In general, I think most of us are fighting to sleep in time, I think. I know I am. But sleep is critical. So lack of sleep can definitely impact the microbiome, especially if it's ongoing. The sleep debt is a problem. People trying to catch up with sleep. But this is important that you guys know how powerful these things are. So you just don't throw your hands up in the air if a patient's microbiome isn't changing with supplements. Because it could be these other factors. Like, do they actually sleep? Right? And then they found circadian rhythms are critical too. Circadian rhythms being when you eat, when you sleep, when you wake up, that being consistent is critical for the microbiome. So if you travel and go back and forth all the time, that really destroys the microbiome, especially in different time zones. You're eating at different times of the day. that has been shown to really have a huge impact on the microbiome, its functions, and also its diversity as well. So in this paper, if you guys get a chance to read it, it talks about the time of eating being the same. Like the best thing to do is sleep, eat at the same time, do your things at a routine basis. When your body gets associated with that rhythm, the microbiome is at its healthiest potential. Okay? And then they show, like, if when people disrupt this, like their time of eating and their sleep, this can actually lead into metabolic syndrome. This is the whole field of immunometabolism, how people disrupt their metabolism and gate obesity from immune mechanisms. And the microbiome is a big part of that. So, very interesting. Because the bacteria in the microbiome determine how you process the energy you eat, whether you store it as fat or not. So when it's disrupted with abnormal circadian rhythms, it can impact the microbiome's ability to properly control insulin and how it uses energy. Very fascinating stuff. Exercise modifies the gut microbiota with positive health effects. Recent studies suggest that exercise can enhance the number of beneficial microbial species, nothing to do with food, enrich the microflora diversity, and improve the development of commensal bacteria. All these effects are beneficial for the host in improving health status. Best scenario, circadian rhythm, eat at the same time, exercise at the same time, exercise every day, don't overtrain. Those things change the microbiome. Pretty fascinating. Would you ever think that exercise would change the microbiome? I mean, it's amazing. Okay. And then if we take a look here, we can look at the other things that, this one here. Now let me show you, this is the opposite effect of this. What they found is that the microbiome also impacts how you can exercise. Conclusion, this present review provides a comprehensive overview of how gut microbiota may have a key role in controlling the oxidative stress inflammatory response, as well as improving metabolism, energy expenditure during intense exercise. So they go to show in this paper that goes, not only does exercise help microbiome diversity, but the more diverse your gut is, the better athlete you can be. Because your microbiome, if it's more diverse, it can process energy much more efficiently. So you have more performance. So this is a paper for athletes, like how do you improve athletic performance? Diversify the microbiome. Okay? And then this is the paper where they looked at glyphosates, pesticides. This is probably part of the reason our westernized areas are such a problem. And for the most part, they gave these mice phosphatate-enriched food, phosphate exposure in their gut, the diversity went down, and these mice all started having anxiety disorders from this exposure. So some of this is just pesticide exposure. I mean, this is what's so frustrating. Like, you can only do so much. You can try to eat well, you can try to sleep, and then you get all this other stuff you can't control, and this is what we all have to deal with, right? That's why we're all working with chronic patients, and we're all fighting for our own health and anti-inflammatory model and best sleep and, and all these things, because we have all these various factors. Intermittent fasting has been shown to help change the microbiome. I know it's very popular, but it's something that really helps with gut diversity and richness of bacteria. So the microbiome not being constantly overloaded with food, having breaks in between, seem to be great for its activity, however it's done. And artificial sweeteners, stevia, all these other natural, they can impact the gut microbiome. So you want to limit those types of things. Now, here's the list. This is the key thing here. Factors that increase microbiome diversity. Diverse list of vegetables, fruits, and fibers. Okay, notice they didn't put diverse list of animal meats. That doesn't make a difference. Bear, salmon, beaver, halibut, they don't change diversity. It's plant fibers. Okay. Diverse meals, trying to get lots of diversity in the meals. Sleep, critical. Normal, consistent circadian rhythms. Exercise, but not overtraining. Outdoor activities, that's the other key thing. Outdoor activities is huge for microbiome, like touching everything and interaction with pets. I was speaking at UCSF last year with a conference, like an integrated medicine conference, and the person that spoke right before me was the main researcher at Stanford that does all the microbiome diversity research. And then we had lunch together in a group with a few other people. And she was telling me one of the things they were doing in their lab was having a competition to see who could have the most diverse microbiome at the end of the month. which then included going outdoors, not showering, pretty much eating dirt, getting exposed to as many bacteria as they could in the environment, but those people that did that won. Okay, so I'm not sure if you need to do all that, but there is some uniqueness of pets have been definitely shown to help with the microbiome, and outdoor activities are important. And all the things that destroy the microbiome are limited diversity in the diet, same meal every day. Lack of vegetable diversity, living in extremely sterile environments, urbanization, lack of sleep, disruption in circadian rhythm, not having consistent sleep and meals at the same time, sedentary lifestyle, lack of exercise, antibiotics and NSAIDs, pesticides, which you can do the best you can for, and artificial sweeteners. So when you get to the point where you're trying to improve the microbiome, trying to improve tolerance, like this is the game. Like you could literally pull these charts out. And for all these charts that you see that have the ones that we made, they're all in your diagram section. The reason they're in your diagram section is so you can print them if you want. And like give it to the patient, have a goal, or, just help you with your patient education. So you might have to go, okay, which of these are you failing on? Which of these are you doing well in? And then just go through each of these lists and then see what you can change, and then have a goal and see what, maybe they can focus on sleeping more, maybe they can focus on diversifying their diet more, maybe they can focus on the circadian rhythm more, but when it comes to, for example, a person who's got autoimmunity, let's say they have MS, and you're trying to control their tolerance, you pull out a list like this and go, your white matter depends on this. What can you do? What are you not doing? Let's make a goal for the next 3 weeks, optimize this list, see what happens, see how you feel. That's critical. And then the supplements to really support microbiome diversity are short-chain fatty acids, butyrate, propionate, acetate, and then fiber sources, guar gum, pectins, flax seeds, cellulose, psyllium seeds. Dr. Bijani, do you guys know Dr. Bijani? He's the guy who's the main chief guy at Cyrex. You know what he does every morning? He takes water, he puts psyllium, flax, other fibers in the water, mixes it, and it's the first thing he drinks. Why do you do that? He goes, because I'm an immunologist. I'm all, okay, good answer. He's doing all that because he knows as an immunologist, if he gets fibers in every day, a diverse list of them, it's going to impact his microbiome. Does that make sense? That's his first thing in the morning. That's what he does. So if the world's leading autoimmune immunologist is doing that, it's probably not a bad idea for people to implement. Okay, now, the next question is, why are we so different? Why are, like some people come in, they've had allergies and eczema and immune issues their whole life, and some haven't. Some are constantly struggling. Some turn 30 and everything goes to hell, and they're 60 years old now and nothing's gotten better. Right? So we all have different starting areas of what happens to us. So first of all, the microbiome is constantly changing throughout our lifespan, and the healthier it is, the healthier we are. But it all starts from the very beginning, breast milk versus formula. So there's a percentage of your microbiome that is going to be your microbiome from the first few years of your life. Whether you had breast milk, whether you were born through a vaginal or a C-section, whether you were given antibiotics, that will have some long-lasting effects. So you can look at some people who were on formula, had a C-section, had lots of ear infections, were on tons of antibiotics. Now they're in your office with chronic allergies and immune issues. They're not the same patient as someone else. I mean, you're going to try to do whatever you can to the best of your abilities with the lifestyle nutrition approach, but they're not going to be like other patients. Okay? So when you look at some of these studies, the study confirms bacterial community in breast milk and tracks. And basically, remember, formula milk is sterilized. And breast milk has bacteria. And these bacteria are critical to help prevent allergic diseases down the road, developing allergic diseases. So I don't know why it's even controversial. This is in the immunology world, it's not controversial. The rest of the world, it's controversial. You have to have bacteria in breast milk to really improve microbiome health. Okay? And then there's another paper, can nutritional modulation of maternal intestinal microbiota influence development of the infant GI tract? And the answer is yes. In conclusion, during the last decade, important evidence has shown that during pregnancy, maternal gastrointestinal tract microbiota or its products may also be transferred to the fetus neonate through the placenta by breastfeeding. So here's the gut. So what they show is like the gut bacteria totally impacts the fetus's bacteria. So when you're looking at the explosion of autism, you're looking at kids with developmental disorders, and you're looking at like, oh, I'm pregnant, I can eat whatever I want, not a good idea. If there's any time to have the richest microbiome is really during prenatal. Next year with the every year we do 3 courses. Next year with cross-nutrition, I think we're doing infertility. I'm going to talk to you about all the research on prenatal, because it's definitely more than folic acid in multis, okay? So like one part of updated prenatal care is to have the healthiest, diverse microbiome before pregnancy. Absolutely critical, okay? Because it changes your child's immune system for the rest of their life, okay? And then this is one paper that was published, decreased gut microbiodiversity, delayed bacteroides, C-section, and they go on to show that C-sections totally changed microbiome potential. And as a matter of fact, right now, you can measure everyone's microbiome, like the bacteria in the gut, and you can tell from the bacterial species whether when they were a child, they were born through a vaginal birth or through a C-section. To this day, any adult. Isn't that amazing? So if you were given a vaginal birth, you'll have different bacterial populations than if you were given a C-section. There's different bacteria on the skin that show up at higher populations. So they can, in a predictive model, look at your microbiome and tell you what kind of birth you came through. So it has long-lasting effects. Okay? So maybe your GI protocol is for prenatal work. It's not even for symptom. You're just trying to get the gut as healthy as possible. And if it was, you would start north and go south. All right. Now, this is a paper there with mice, because with mice, they don't have a lifespan. They can really check to see their offspring and their offspring and offspring, and altering intestinal microbiota during a critical development window has lasting metabolic consequences. So when they disrupted the microbiota in mice, their children, their offspring, all had problems. Okay? Now, so that's one thing. So we have to know is that the key thing with this, before I move on, is there's different susceptibilities for microbiome and gut issues based on what's happened to them. Okay? So you could have a patient that comes in and has the worst gut, and you're thinking, God, they must have an infection or something. And then when you actually do their history, you see it was a lifelong issue. It's the typical story. C-section, formula, ear infections, multiple antibiotics, immune stuff their whole life, constant going on, and now they're in your office. You're not going to reverse all that. You're going to try to do everything you can and see how far you can get, but they're always probably going to have some immune susceptibilities. Their immune system is now completely different. Okay, so it's also a good idea as you do your histories with patients that have GI issues to go back and go, hey, tell me about your childhood and your gut function, your immune function, and then you'll see the list with some of them that it's pretty obvious that they're susceptible for all immune issues now because of that. And then if you have them, you do what you can. Okay, so health and disease. So all these things, by the way, on the top of this chart are all the things that impact the microbiome. And you do the best you can. Lack of fermented foods, stress, pesticides, herbicides, medication, fungicides, GMOs, hormones, and livestock is a big deal. That is totally messing us up, just so you guys know. It's making us antibiotic resistant. It's totally changing our diversity. So when they put these things in milk and in livestock, these antibiotics, that's part of the reason we have less diverse guts now. Okay? They're really doing it so they can not have animals die and they can make more profits, and it's making everyone's microbiome significantly less diverse. And then when these things happen, you can get health issues, but I really want to move on to over here. This is the gut. And you see this pathway here, microbiota, normal microbiota on the left-hand side. You see structurally altered microbiota. You see how many colors are here? That's representing diversity. More colors here, more microbiota diversity. Only one color here, less diversity. So this was a paper that was published in Journal of Clinical Investigation, and they were looking at insulin resistance and obesity. What they found was when there was lots of microbiome diversity, that these bacteria produced high amounts of short-chain fatty acids. And these short-chain fatty acids produced by all this gut bacteria diversity impacted what are called G-coupled proteins and adipose lipase tissues, which modulated how they burned and used fat and how they managed insulin levels through AMPK. So this is where the majority of research is happening now in diabetes. When there's lack of diversity, there's this problem with the gut. Okay? Now, initially, fasting helps diversity. Like intermittent fasting helps diversity. And then there's a point it goes counterproductive because you have to have plant fibers. So like multiple diverse things, fasting, those all kind of work to keep the tract going. Lots of religions have fasting in different periods. It's really a beneficial health approach to them. And, sometimes when you have a person who's got insulin-resistant metabolic syndrome and you can't get their insulin levels down, you may have to forget all the insulin receptor stuff and go right to the gut and make a difference there, okay? Now, when you look at some of the studies they did in animals, you guys have heard of fecal transplants? So in animal studies, they take the microbiota from fat mice, put in skinny mice get fat, and vice versa. Take fat mice, give them skinny stool samples and fecal transplant, and they lose weight. They did this in humans a couple of years ago in Germany, 30 subjects, diabetics, obese. They did a fecal transplant with skinny individuals. It worked. They all lost weight. Within 6 weeks, they all lost a tremendous amount of weight. No change in their diet, just from a fecal transplant, and all came back. That just shows you can't use it as a treatment because they were going to open up like fecal transplant centers of America everywhere. But it shows you the microbiome is a key factor in how that's done. So you can see different species. They first thought it was this Bacteroides, Ficteroides like ratio. That's shown to be much more different than that. It's more complicated than that. The other thing to know is during pregnancy, the microbiome actually changes to the microbiome of an obese patient, normally. So the mother can gain weight, support their fetus. That's the only time, like a normal, healthy, skinny woman with their hormone surgeries during pregnancy, the hormones change their microbiome. They sample it will look like a microbiome of an obese individual. And then their gut changes. The key thing, though, is you want their microbiome to change back. And some don't. And they'll say, after my pregnancy is never the same. Okay? So they'll continue having the microbiome of an obese patient. This was an interesting paper. This was my study. They looked at the microbiome diversity versus non-diversity. Germ-free means they sterilize their gut. There's no germs. And SPF is basically a normal gut. And they looked at motor development. And the colors you see is motor development during a mice developing in their brain. So when there's less microbiome diversity, there was less motor development throughout the brain. So now think of what that means to a kid with a learning disorder. What does that mean to a mom with a microbiome issue transporting to the fetus? So picture this. A mom doesn't eat diverse foods, is already sick, has endotoxemia, gets pregnant, has a kid. Their kid has no microbiome diversity, so now they're born. Now they get a C-section. Now they get antibiotics. Formula. That whole cascade completely changes how their brain will develop. Okay, and then you could add other things on top of that. Okay, so then we know that the microbiome is also involved with behavior. So they take mice, they give them antibiotics, they see personality changes with them in some studies. There's a study they did where they took a group of 2 different species of mice. They took the BLB mice, it's very timid, and they took the Swiss NIH mice, which is very courageous and doesn't care, will just walk around the middle of the room, do whatever. And they gave them fecal transplants, and their personalities switched. This is amazing. So the ones that were courageous started to become nervous. They started to hide in corners. The ones that were hiding in corners started walking all over the place. And that just shows you how powerful the gut microbiome is in our brain function, our mood, our personality, right? They did the same thing. They did another study where they gave a pathogen to the drinking water, and these mice that used to be able to figure out the maze were no longer able to figure out the maze. Just from bacterial exposure in the water. In most of these studies now, and in brain MRI studies, are showing high correlations with inflammatory bowel disease and brain issues. We know we get blood-brain permeability issues. The microbiome totally impacts those areas. So in addition to mood, it causes brain inflammation. And now there's also an explosion of research that is showing the microbiome can impact atherosclerosis. And there's certain bacteria in the gut that if you have high amounts of, which are listed here, this is a great diagram from Dr. Vijdani. And then if you combine these bacteria with foods, you actually produce a compound called TMAO, and TMAO directly destroys blood vessels. And they showed these with, and what they showed is, like, you see how meats and cheeses have been associated with heart disease? They go like, red meats are associated with heart disease and so forth. That's in people that metabolize red meat, L-carnitine, to these postbiotic substances called TMAO, which then causes blood vessel destruction. So it's not the red meat alone. It's the red meat plus the bacteria in their microbiome. So what they're saying is, our model of like looking at these studies where we just look at red meat can be completely changed in the future if we can look at their diet with their bacteria, A plus B equals C. And then this is one of these studies they showed here. So these are people that have these bacterial growths that can convert meat into TMAO aggressively. And here's chow, here's carnitine. Carnitine is what's in meat. You can see this is blood vessel, this is atherosclerosis in red. Okay, normal chow. They give them lots of red meat, carnitine. You see the atherosclerosis develop. Same thing, they give them the chow plus antibiotics to kill the bacteria that they found that was causing the conversion of carnitine to TMAO. And you don't see the atherosclerosis. And this was done just in this cross-sectional study to show that your gut bacteria can impact your atherosclerosis. Interesting, right? So what does all this mean? This is all interesting and fascinating stuff, but at the end of the day, clinically, your goal is to improve the microbiome so you can fix these different pathways. And improving the microbiome has to go through a set of steps. Now, there's some overlap. Like, for example, you can treat leaky gut and still try to diversify their diet at the same time. You might need to do that. Like, you might not be able to heal someone's leaky gut that's chronic, has autoimmunity, until you follow all the strategies like this, right? So this is a combination approach. But you're going to have a hard time just fixing microbiome diversity if you have digestive issues or if you have ongoing inflammation, okay? Jumping to the microbiome with like a probiotic of whatever kind to try to make the biggest change is not working north to south, okay? So you always want to work north to south. This is a sheet you should probably print up and like go with your patients and go, okay, let's talk about each of these things. Where are you at? This is one of the key things you want to assess. What's not working, and let's check up on you in 3 weeks. And that's the kind of the big picture, and then you can give these support products. Okay, so what we've done so far is we've gone through digestion, all the major things that can go the way north to south. We talked about intestinal permeability, different types, and endotoxemia, and then we talked about the microbiome, all the ways to change diversity. Is everybody okay with that? Okay. The hierarchy is critical, and taking a step back and knowing the big steps involved with each of these and developing a plan is the way to do it. You don't want to just give everyone the same protocol. Let's take a break, and after the break, we'll take questions, and then we'll do a review. Okay? Great. Thank you. Thank you.